Hello, everyone, and welcome to today's webinar, Understanding the How and the Why, Biosafety in Centrifugation. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Bob Rodriguez, Sales Specialist at Beckman Coulter Life Sciences, and Dr. Lori Kelman, Professor of Biotechnology at Montgomery College. Welcome, and you may now begin your presentation.
Welcome to the talk, Understanding the How and the Why, Biosafety in Centrifugation. This document contains proprietary information owned by Beckman Coulter and is not for distribution without express written consent of Beckman Coulter. All products referred to are for research use only unless specified for IVD use. Beckman product and service content are trademarks of Beckman Coulter. All other trademarks are the property of their owners. The views expressed here by Lori Kelman are her own. Lori has not received compensation for this webinar from Beckman Coulter. Centrifugation is an indispensable laboratory application for separating and purifying a range of biological materials. Although a cornerstone, gaps in general understanding of good laboratory practices, 21 CFR Part 58, are not uncommon and can serve to impede efficiencies, negatively impact research goals, and can even increase laboratory safety risk factors. Lori?
Hi, I'm Lori Kelman, a professor of biotechnology at the Germantown campus of Montgomery College in Maryland. We prepare people for jobs in the local biotech industry by providing hands-on training in cell culture, protein, DNA and RNA methods, as well as introducing students to the basics of GLP and cGMP in our biomanufacturing suite.
And I am Bob Rodriguez with Beckman Coulter Life Sciences, a sales specialist on a variety of products, including centrifugation. The intent is to provide information to assist with utilizing centrifugation in a more productive and safe manner.
These are our learning objectives for the talk, and we're going to review the fundamentals of centrifugation, talk about the basic use and maintenance of centrifuges, and discuss centrifuge safety in general. I'll be talking about the how, or probably what I'd like to call Centrifuge 101, and Bob will be telling you more about the why.
The first part of the talk includes fundamentals of centrifugation.
Lab centrifuges are not used in isolation. If we work our way out from the center of this diagram, samples are held in tubes or other containers that have to be selected based upon the appropriate rotor for the application. The centrifuge itself is located within the lab environment, and all of these choices, from the tube selected to the management of the laboratory, can have consequences for safety.
Centrifugation is a technique for separating particles in solution using centrifugal force generated by a spinning rotor. Under this force, directed outward from the center of rotation, particles are separated by size and density, with larger and/or denser or higher mass particles moving faster. This force is opposed by a buoyant force because these particles are displacing solution volume as they travel, and by frictional force depending on how much particle surface is interacting with the solution. Each of these interactions are related to analyte, mass, volume, and shape. The most popular method for separation by centrifuge is by pelleting, where all or most of the particles are collected at the bottom of the tube. Simple pelleting collects all particles, and differential pelleting stops the run while the smallest subpopulation is still in suspension. This is done when the smallest particles are being retained for downstream work. Note the picture of the conical tube with cells collected at the bottom using simple pelleting. And note the diagram at the right showing 4 time points where during the run leading to time number 4, where the run is stopped in order to collect the smallest fraction still in solution, which has been separated from the larger fractions which have pelleted. These are 5 centrifuge run and environmental conditions which affect the separation and safe operation. When using a centrifuge, rotor speed and imbalance potential are always a safety consideration. Ambient temperature will affect the centrifuge's ability to cool well. Deceleration profiles are related to the degree of unwanted mixing which can occur when the brake is applied.
And centrifugation produces aerosol. So depending on the hazards that are specific to your samples, you may have to take more than the regular precautions.
There are 2 common rotor types, swinging bucket and fixed angle. The rotor pictured on the left is a swinging bucket rotor, where the bucket swings to a horizontal plane during the spin. All particles move down the length of the tube, and when pelleting, collect at the bottom center of the tube. Separations in a swinging bucket take longer because the travel distance taken by particles is longer than with a fixed angle rotor. The swinging bucket rotors cannot be spun as fast because they are top heavy and hence more unstable. Fixed angle rotors hold the tube at a set angle from vertical. Particles move outward diagonally across the tube, hit the outside wall, and slide down and are collected at the bottom outside of the tube. Travel distance is less, so run times are less. And fixed angle rotors are more stable, so they can be spun faster, which decreases run times even more. What tube to use has more options than any other centrifuge considerations, and some of these features relate to safety issues. Volume to be processed and product to be collected are key considerations performed by every user. But tube material, reuse of tubes, proper adapters, and the ability of the tube to contain spills all affect biosafety and should be considered.
Part 2, basic use and maintenance. Your centrifuge must be correctly installed. And if you move a centrifuge, it has to be reinstalled and re-leveled. Benchtop centrifuges should be located away from the edge of a counter, because if it should move during a run, it shouldn't also fall off the bench and cause a much more serious incident. Load the centrifuge correctly. Check that the latch is properly closed by listening for a click with some models and by rechecking with another push on the lid. When operating a centrifuge, remain near the centrifuge as it comes up to speed and pay attention for anything out of the ordinary. If you hear an unusual sound or if you feel an unusual vibration, shut the spin down. After a run, inspect the inside of the centrifuge and the rotor and clean both. Cleaning is a critical step in centrifuge maintenance, and once this becomes a habit, it's really easy to do. I worked in 2 different labs where I was considered a mechanical genius because everybody thought I could fix any piece of equipment, but I have to put air quotes around fix because all I really did was clean the equipment. Once a centrifuge was, air quotes, broken because the lid wouldn't latch and, it wouldn't turn on. A really quick visual inspection showed a piece of tape rather obviously stuck in the latch mechanism. I used forceps to remove it and, air quotes, fix the centrifuge. The previous user of that centrifuge should have noticed a label missing from one of the samples and followed up and found it, but obviously they did not.
I must add, Lori's story is a simple but real-world example of how common sense and a little care can eliminate many problems. The gravitational forces generated by a spinning rotor can range from around 1,000 to over 100,000 Gs. 1 mil of water weighs 1 gram at 1 G, but 10,000 grams at 10,000 Gs. As a result, imbalances are magnified, leading to imbalance being one of the biggest causes of accidents. Under these stresses, rotor materials, whether made of metal or carbon fiber, are stretched every time they are run. Other common causes of rotor accidents include little or no inspection of the rotors for stress signs like cracks, sticking of buckets due to insufficient cleaning, no or damaged O-rings, and weak or improperly sealed tubes. Proper tube sealing is especially critical when containing infectious samples.
Depending on the biosafety level of your samples, you do have additional options, such as sealed rotors and filtering the centrifuge exhaust. My centrifuges weren't used for the past several months, and Bob reminded me before we recorded this that I'd really better take a good look at my O-rings. Tube selection is complicated because there are so many options. Make sure you're using a tube appropriate for the intended use. The tube should fit the rotor. And this sounds obvious, but the force of centrifugation doesn't allow for gaps or movement with ill-fitting tubes. The shape of the tube needs to fit the shape of the rotor cup. Each tube should be inspected by eye before you use it, and there should not be any visible cracks or stress marks. Notice the red arrows in this diagram because that's the direction of stress for a spinning tube, and that could be where you'll see a crack. And if your rotor isn't clean, a particle left at the bottom of a rotor can cause a star crack on the bottom of a reused tube. The caps of some tubes have multiple parts. Make sure that you have all the parts and you use them correctly, and make sure when you're balancing that all the parts are present. Deformed tubes or caps should not be used. And the best practice is to discard centrifuge tubes that can't be used in a centrifuge. When in doubt, throw it out. You've got too much junk in that drawer anyway, and leaving those tubes around really just asks for somebody to use a tube that shouldn't be used. Know the G limit for the tube and do not exceed it. Consider solvent compatibility and make sure the tubes that you use are made of a compatible material. If you need to sterilize the bottles, use the correct procedure. I'm sure Bob can tell you stories, I know I can, about collapsed and deformed bottles due to incorrect autoclaving. And properly fill tubes or bottles. Never overfill them. Some of them have to be filled to work, but don't overfill. Split your sample into more tubes if necessary.
I also, regarding tubes, have a quick tube story. One of our ultracentrifuge rotors spins 1.5 mil microfuge tubes at speeds that generate up to 186,000 Gs. Several times I have received calls from users complaining that all of their tubes were crushed during a run. I had to tell them that only our microcentrifuge tube is rated for forces up to 186,000 Gs. All other tubes that I know of are rated for forces no higher than 30,000 Gs.
Balancing tubes is really important, and here's where your laboratory environment matters. An electronic balance should be conveniently located near your centrifuge or the sample prep area. You shouldn't have to walk all over the lab to find it. Weigh your tubes with the caps, and also weigh them after carefully wiping off any ice or liquid from the outside of the tube. A rule of thumb for balancing tubes is they should be within 0.2 grams of each other. Wipe the tube off again before you load it into the rotor. And I know this should be obvious, but ice crystals and water have mass, and ice crystals melt into liquid that can dirty your rotor. Loading the rotor involves placing balanced tubes diagonally and directly across from each other. For fixed angle rotors, this is often pretty straightforward, but the image at the bottom of this slide shows swinging buckets with multiple samples, and these can be confusing to load. Remember, I teach people who have not used centrifuges before. You want to draw a straight line from one tube through the spindle straight across to the balance tube. Notice the incorrectly loaded mirror images here. A properly loaded bucket, they're not mirror images. Best practice is to have another person check the loaded rotor before you start the run, and it is never a waste of time to ask if you have a question about safety. Do not start a centrifuge if you're not sure that your tubes are balanced and the rotor is loaded properly. It's also best practice to spread the load evenly around the rotor rather than concentrate it directly across from each other.
Every rotor is designed to be spun at a maximum speed, but after years of use and stress, the rotor material does not have the elasticity of when it was new. Older rotors should be inspected either by trained lab personnel or by the rotor manufacturer. In general, the maximum speed of old rotors should be reduced or derated by 10%. This is related to the run speed selected and whether that speed exceeds what is safe. Note, though, that higher speed centrifuges have overspeed detection features to prevent rotors from exceeding their speed limits. But it is still good practice to follow rotor speed limits, especially with older rotors. Another safety-related practice is to be aware during early rotor acceleration of excessive vibrations, which is a sign of an imbalance. If this happens, stop the run. Centrifuges have imbalance detection features, but at times do not pick up imbalances quick enough to prevent a costly accident.
When I start a run, I put my hand on the centrifuge to feel for the vibration, and I also listen to the centrifuge as it comes up to speed. There's a normal vibration and a little bit of a wobble around 700 or 800 RPM, but then the vibration settles as the speed increases. If that does not happen, or if you hear an unusual sound, shut the run down immediately. I learned a new thing while preparing for this. Bob told me the name of that vibration or wobble, and I've forgotten what it's called.
It's called precession.
Precession, right. And it's normal on a centrifuge, but if it gets worse after that, that is not normal.
Lori and I have discussed why centrifuges are used as a separation tool, but here I will outline why there are many centrifuge types. I've explained that centrifuges separate based on size and density or mass. G-force causes particles to move, but large and heavier mass particles move faster. In biology, mammalian cells are relatively large at approximately 10 microns in diameter. They can be pelleted at approximately 2,000 Gs in a few minutes, so a low-speed bench centrifuge like the one pictured above on the left is sufficient. However, smaller particles like bacteria and subcellular organelles move slower, so they need higher speeds and forces, typically in the 10,000 to 50,000 G-force range, to pellet them in approximately 10 to 30 minutes. The centrifuges in the middle above are used for these applications. Very small particles like viruses move the slowest, so they require forces typically over 100,000 Gs, generated by an ultracentrifuge, to move fast enough to perform a separation in approximately 2 hours. So the size and or mass of a particle to be separated dictates the best type of centrifuge to be used.
Part 3, safety considerations. When people teach centrifugation, we often use the example of the spin cycle on a washing machine. And while that uses centrifugal force to remove water from your towels, it's really a terrible example because the force involved is so much less than what we use in laboratories. I teach new centrifuge users, and it's my observation that many high schools have tabletop microcentrifuges, the little ones that spin 1.5 mil flip-top tubes. Even these don't generate the forces involved in floor model centrifuges. The forces involved in centrifugation, it's not within our experience, and so it's really difficult to imagine. Be sure that you get training, because what applies to a microfuge, like balancing tubes by eye, is not how to work with other centrifuges. What this slide is showing is that the cap of a pen, if subjected to 1 million G, it would effectively turn into the weight of a small car. That's the kind of force we're talking about. And that's why balancing and loading a centrifuge is so critically important. And this is also why a centrifuge, which might not seem to do a lot except make a little bit of noise while it's running, can cause the occasional catastrophic laboratory accident.
Many rotor accidents are caused by user error and hence are avoidable. And as just discussed, the forces generated in a centrifuge can create considerable damage and even an unsafe event to those near the centrifuge. Above on the left is a picture of a rotor accident in an ultracentrifuge where the rotor made of titanium was obliterated. Proper centrifuge, rotor, and tube care can greatly reduce these incidents. However, note that this particular incident occurred over 30 years ago. Ultracentrifuges today have better imbalance detection features to prevent many of these incidents from happening. The picture on the right shows corrosion highlighted by a dye in an aluminum fixed angle rotor. Aluminum is the most common material used in low and high speed rotors. Routine rinsing of salt deposits accumulated in the rotor wells as part of a regular rotor maintenance can prevent this and ensure safer operation.
I've never been in a lab at the moment where there was a catastrophic rotor incident, but I've been in several labs after there was, and I will agree with Bob, this was decades ago. In one case, the rotor was old and the lab knew there was a chance that this would happen eventually, and it did. That particular lab did a lot of ultracentrifugation, and it did not derate their rotors, but used them to failure, again, knowing at some point that the rotor would fail. Another incident that I'm aware of was due to operator error. The samples were not properly balanced, and in this case, the centrifuge was lost. And of course, the samples were lost at the same time. I was also in a lab where a balancing error led to a spindle on the centrifuge that stretched out basically into a very, very long piece of wire. We were not exactly sure how that happened. I'm sure Bob could explain it. And I've personally witnessed a number of runs that had to be aborted. All of these were due to either balancing problems, failing to seat the rotor correctly, or failing to check that the swinging buckets could swing freely. In other words, they were all due to operator error.
In the United States, federal agencies provide regulatory guidelines on safety, but interpretation and compliance falls on the individual organization and requires proactive engagement in order to maintain compliance. The role of the organization's EH&S group combined with the individual lab is to train for and enact good practice, and to refresh and review these practices regularly. Thus, in the U.S., regulatory compliance is comprehensive, but falls across multiple organizations, whereas in other regions of the globe, such as Europe, the approach is to maintain all regulatory comment under one organization. However, at this time, that regulatory comment is incomplete. In a biologics development or production setting, safety is embedded into the operation through various risk mitigation activities such as regular centrifuge and rotor maintenance and routine training. The centrifuge and rotor maker builds safety into its operation by considering the centrifuge, rotor, and labware as a system from early design through manufacturing, with the primary objective to ensure safety even in the event of a severe accident.
If you work in labs long enough, there's a chance that you're going to witness or hear of centrifugation incidents. I've described a couple of them already. In my experience, nearly all of them were due to human error. Also, in my experience, in none of them was anybody injured. And that's really a testament to the centrifugation industry, and part of why I think centrifuge safety isn't really front of mind. But losing a centrifuge, you're going to lose your sample, it's going to stop work in the laboratory, and it's a very expensive incident. And So we should all be aiming for 100% centrifuge safety all the time.
The Code of Federal Regulations, CFR, codifies the rules issued by the agencies of the federal government. Section 42 CFR 73 relates to the possession and use of various viruses, including SARS-CoV, where registrants must develop and implement a written biosafety plan to be reviewed annually, describing the biosafety and containment procedures. Part 73.12 calls in part specifically for, quote, centrifuge safety containers, unquote, to protect lab personnel and the public. The International Electrotechnical Commission, or IEC, sets standards globally for the performance and safety of electrical products. Lab equipment and manufacturers like those that make centrifuges belong to this commission. A section within these standards deals with centrifuge lab safety, where guidelines are set for the testing of new centrifuge and rotor systems.
Another thing I learned while preparing for this presentation, I didn't know that there was an IEC. I had no idea, so thank you for that. I really want the course on how centrifuges are tested, Bob.
I might also add they are literally tested and exploded in a blockhouse. So, and this is per IEC guidelines. The Centrifuge Lab Safety Guideline, and here it is, IEC 61010-2-020, calls for the testing of the ability of a new centrifuge to contain a severe rotor accident, which it terms a maximum credible accident. And I might add that in this case here, the manufacturer must take its most explosive potential rotor and literally damage it so that it comes apart during an overspeed run to be able to test the ability of that centrifuge to contain that explosion. The passing of this test means that the centrifuge prevented or contained very small particles from falling over 1 meter from the centrifuge. And acceptance of this test data leads to certification by safety organizations such as Underwriters Labs, CSA, and CE. Beckman Coulter has been a stakeholder in the IEC for decades.
One really important practice is whenever you start a centrifuge, you should know exactly how to shut it down. Press the stop button, turn the speed to 0, turn the time to 0. You need to know how to do that before you press the start button. Panicking is bad when something goes wrong. And once you do that, step away from the centrifuge. I'm glad to know it probably will be contained, but better safe than sorry. On many research lab protocols, the centrifugation step is just this short one-line instruction, and I think that gives the false impression that the centrifuge step is going to be quick. In addition, when your sample's spinning, that's often a chance for you to finally have a snack or take a break or do something else. And in my observation, getting samples into the centrifuge is often a rushed step. Please take your time. An incident may not only cause you to lose your sample, but also to lose a really expensive piece of lab equipment. And of course, there's also the chance of injury. Check and recheck as you balance and load your centrifuge.
Tube leaks and rotor accidents often occur because attention has not been given to the condition of tubes and rotors. Check tubes for cracks and for proper seals if they are sealed. Tube and bottle cracks, especially if they are reused, often start at the bottom and either run around the circumference at the bottom or up the sides as shown in this slide. Use manuals or manufacturer website or product support to learn good practice for rotor balancing, where regular lubrication is needed, how to clean and disinfect rotors. Rotor imbalance allowances can be as low, as Laurie has indicated, as 0.2 grams for some ultracentrifuge rotors and up to 10 grams for large volume high-speed rotors. Accidents can also occur when rotors are not attached securely to the centrifuge, or when the rotor is missing a critical part, such as a lid. I must add also O-rings. Missing O-rings is another cause for all kinds of different centrifuge problems. Request that the manufacturer inspect all rotors to identify rotors that should be retired due to defects.
I'll add a couple of my observations. Knowing, for example, that your bottles can be 10 grams apart doesn't mean that you should be aiming for that. Remember, if any liquid is on it or ice crystals, that's going to change the mass of it. So please always be closer to zero difference rather than saying, yeah, it's probably okay. That word probably okay really makes me worry. In addition, if you're cleaning your centrifuge on a regular basis and inspecting it, you're going to notice those O-rings that maybe came loose during a run. When safety is a priority, a worker won't feel that they have to rush through a protocol. The lab should be set up with best practice in mind. For example, having a convenient place to balance tubes. Budget enough time for the work to be done and take the time to do things right. Carpenters say measure twice and cut once, and I'm going to say balance twice. Well, I don't really say that, but be sure and be confident before you load a rotor. If you aren't sure which tubes are the balanced pairs, weigh them again. If you aren't sure how to use a centrifuge or any piece of equipment, ask. And if you forget, ask again.
Safety is the result, as we've heard here today, of well-designed and maintained tools such as centrifuges, combined with good practice to reduce risk. Oversight bodies provide guidance, but it is the responsibility of the organization to implement, monitor, and maintain safety standards. For this reason, safety is not applied universally, but is a practice customized for each organization.
Thank you for your attention. We're happy to take your questions.
Thank you, Bob and Lori, for your informative presentation. We'll take a moment for our speakers to grab a sip of water, and in the meantime, we would like to understand our audience better. I will now share a series of poll questions, and your participation is greatly appreciated. Our first question is, would you like Beckman Coulter Life Sciences to conduct a free rotor inspection? I'll give you about 15 seconds to respond. Next, does your organization have an EH&S department who might be interested in learning more about biosafety in centrifugation? Next, I would like to feel a little safer about biosafety. Please have my local BEC LS representative reach out to me to discuss options. And the last poll question, what webinar topics would you like to see from Beckman Coulter, i.e. product applications, techniques and methods, research areas, industry standards, etc.? Please be specific. All right. Well, we'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what is the best course of action I can take to assess centrifuge safety in my lab? Is this process something that Beckman Coulter can help us with?
Hello, can you hear me?
Yes.
Okay, great. Yes, thank you. That really is a conversation that should be had depending on the types of centrifuges and the types of rotors that you use. As mentioned during the talk, centrifuges range from very low speed benchtop models all the way up to floor model ultra centrifuges. The forces vary greatly. The types of rotors vary greatly. Types of labware vary greatly. So really that's a discussion that should be had between someone like myself or someone within the company and the lab.
All right, thank you. Next question. How do you inspect a rotor and how do you clean it?
I'll continue that. Laura, you can enter in if you have a comment.
I was just going to say I mostly just look at it and clean it with water, but I know you'll do a better job of answering this.
Yeah, in general, metal rotors should be rinsed out with DI water with a soft brush. The issue with rotors often is salt deposits at the bottom of the well, which cake up and may cause corrosion problems with aluminum rotors. So our recommendation is to watch that and to remove that salt through this rinsing process. And then at the end, after a good flush, turn the rotor upside down to let it air dry. So that's basic rotor maintenance right there. But also good maintenance or good ideas on, or rather a good procedure on how to treat every rotor. Just keep them clean.
Yeah, and I'll add to that about the laboratory environment. If you've got the sort of rotor where it would really help to have a flashlight to be able to see if you've got some white salt deposits at the bottom, then it would be really nice to have that flashlight handy.
Correct. I might add also that look for small cracks, especially with swinging bucket rotors where the buckets hinge onto the rotor body. Look for little micro cracks. Certainly the first signs that the rotor is starting to age and should be replaced. So that's especially in swinging bucket rotors, but also in fixed angle rotors. This is where a good light will help to shine down into the well and look for any areas where cracks may be occurring.
All right, great, thank you. Our next question, what about pan balances for balancing? Is that okay?
I'll start with that one. Personally, I'm not a fan of pan balances, but for centrifuges that are not ultra centrifuges, they're really fine. It's just in my experience, sometimes they stick a little, and I'm never 100% sure they're working the way they should. I don't know what it is. I think I like the comfort of seeing the numbers on the electronic balance, but I think that's personal to me.
Yeah, I might add also that we have tolerances for each type of centrifuge. So really the balance requirements as spoken during the talk differ depending on the type of rotor, how fast it's run. And so that's going to dictate how careful you should be with balance. But balance is a big issue. Balance is, along with human error, probably the biggest reasons why centrifuges fail. Okay.
Next question. Do you clean a rotor after every use?
So I inspect it after every use.
I don't truly clean it after every use, but if there's any reason it needs to be cleaned, it gets cleaned. And that's the moment to do it because it's going to take a little while to dry. So if you wait to clean it until right before you're about to use it, you can't use it right away.
I would add that shouldn't have to do that. That's adds a lot of time to what needs to be done in the lab. It really, I think the eyeball there can tell you whether that needs to be done because tubes leak, bottles leak. You can often see that there's been a spill or even a minor spill. So that might require a quick rinse in a rotor to take care of. The other telltale sign is what's at the bottom of the centrifuge chamber. If there's been a bad leak, you'll see it. So, of course, after something like that, wipe that up. But I would say good practice is just to keep an eye out for when that is needed, but you shouldn't have to do that every run or even, I think it's better just to set in a regular practice. Every week, for instance, check on the various things that need to be checked.
All right, thank you. Next question. Is there somewhere that I can find information on chemical compatibility and O-rings?
Yes, at the back of our rotor catalogs, we have chemical compatibility charts. Certainly, you're welcome to contact me or those in the company who can help you there. O-rings are specific to each rotor. So again, the catalog is the best place to go to the rotor page, the particular rotor page of interest, and it will contain the O-ring information. Keep in mind also, there are often multiple O-rings with a rotor. So just make sure you are looking at all the O-rings that need to be replaced.
All right. Next question, can I just use volume of the same solution in the same type of tube to balance instead of weight?
So I'm going to say no. For a microcentrifuge, a tabletop microcentrifuge, sure, balancing by eye is probably good enough. Once you get to something that's going to generate more force, please balance by weight. I know some people who do work with things like deuterium, and balancing by eye, if you've got water and something with heavy isotopes in it, that's not going to work. And Bob, you probably have something to add to that.
Yes, and during the talk, Laura and I both talked about how G-force multiplies the mass of what you are spinning. And that's the thing to keep in mind, is how much mass is being spun at the forces that are being applied. And then think of that as a multiplier. So any differences between one side and the other side of the rotor are multiplied by the amount of G's that are being applied to that spin. So that's really the thing to keep in mind, is what is that difference, and minimize that difference, certainly to within tolerances, but even better than that if you can.
All right. This is more of a comment that maybe you would like to add to it. In clinical labs, it is a good idea to keep a log. Do you have any comments you'd like to add to that?
I do. Just when I was listening to what we just said, I realized we forgot to mention logbooks. You can't derate a rotor, for example, if you haven't kept a logbook, because you won't know how often, how much you've used that rotor. Yes, I'm a huge believer in logbooks. And if you're going to use a logbook, everybody has to use the logbook. You can't go, well, this is only a 5-minute spin. So yeah, I'm a fan.
Yeah, the thing to keep in mind with rotors, and again, this was mentioned during the talk, that during a spin, the rotor actually stretches, and at the end of the spin, it relaxes. And that process is called a cycle. So our concern is how many cycles has that rotor been exposed to during its lifetime. And that's what the logbooks are all about, is trying to keep track of how many cycles to understand when that rotor should be replaced. because the rotors don't return. Their elasticity is not the same when they are old as when they were new. So as a result, over time, that rotor is slowly deforming, and so that's why the cycle count is important. However, the number of cycles and how to track that is often tough, as busy as labs are. So what we often do is we try to estimate how many cycles per month, per year, and then during its lifetime. And that's what gives us an understanding, combined with the age, of whether that rotor should be replaced.
All right, thank you. Next question, we are looking to have an OQ performed for our centrifuge to assess speed. Do you recommend an OQ be performed annually, or would there be another annual service besides the routine PM?
I would again point to the regulatory requirements for your lab, your department, your organization in this OQ setting. If it's a GMP setting, often there are internal corporate timelines that need to be met with regard to GMP SOPs. So that really is specific to the organization. An OQ for a centrifuge calibrates speed, time, and temperature. And it really is something that does not need to be done very often. My suggestion would be maybe 6 months, possibly a year, again, depending on corporate requirements. But that's something to discuss internally as to how often that needs to be done. The only time I would say an OQ needs to be redone is when that centrifuge is moved.
All right, thank you. Next question. I am having trouble getting small pieces of hay, approximately 1 millimeter ground, to form a solid pellet in water. Do you recommend increasing the time or the force first when coming up with new centrifuging conditions?
So I'll take a stab at that. The answer is I don't know the answer, but as someone who used to work on plants, I'm wondering if your hay isn't buoyant, if it either doesn't have some trapped air in it, or it's just less dense and not going to pellet so well. You said small. It's going to depend on what you're trying to do there, and I'd be happy to try to answer you more personally by email if you want. Did you have any thoughts on that, Bob?
Yes, and you hit it, Laurie. It really, the separation is relative to the difference in density between the particle and the solvent, which in this case is probably water. So if a particle is close to water in its specific gravity, it's going to be a difficult separation. And there are times with an oil, lipids, for instance, and on the clinical side, classic example of lipids being less dense. So in a centrifuge, lipids will float, and actually that's taken advantage of in various centrifuge separations.
Yeah, if you want those, if you want the little hay particles, you may wind up having to do a buoyant step gradient kind of centrifugation step.
Mm-hmm.
Okay, thank you. Next question, what will be the general RPM of the rotor? Why is 0.2 G so important?
Okay, I'll take first shot at that. 0.2 is tough, I know. It's not much of a difference, but again, keep in mind that 0.2 relates in usually to swinging bucket rotors, ultracentrifuge swinging bucket rotors, which have forces well over 100,000 Gs. So that 0.2 times 100,000 now is significant. So that's what we're saying is it really depends on the force applied and combined with that differential, that weight difference. And that will tell you how important it is to be able to balance each side. So, and again, I made the comment during the talk that swinging bucket rotors are top heavy, so they are less stable. So that also adds to why that tolerance is so low. But that 0.2 doesn't relate to many of the lower speed rotors. So you'll find again in the manuals or by talking to us what that tolerance is. It may go to a gram or even higher depending on what the speed is and what the rotor is.
All right, thank you. Next question. Can you use 70% alcohol to clean the centrifuge chamber?
So the thing about alcohol is it is a disinfectant. So if your goal is to disinfect, you could use that. It's also a different solvent than water. My recommendation, and Bob will correct me if I'm wrong about this, is you start with water. I'm assuming most of what you're doing is aqueous. So your first solvent's going to be water. So water, possibly followed by the 70% alcohol, and then maybe you're disinfecting. The other thing the alcohol will do is it will help things dry a little faster. But in that case, I'm calling it just kind of speeding up the drying step. Mm-hmm. Anything to add, Bob?
Yes, and again, I see it just like you do as two steps. First is cleaning, then disinfection if that's a requirement. 70% is as high an alcohol content as I would recommend. That's actually what our recommendation is to disinfect. Bleach is also another disinfectant, but that's, I would say, I think it's a 5% bleach solution that we recommend there. Keep in mind though, bleach will bleach your rotor and it will whiten it over time, but that's not a structural problem. It's just more of a cosmetic issue. But 70%, if that's what you're using as a disinfectant, is fine.
Yeah, and I'll add, anytime you use 5% bleach, you have to then clean with 100% water to make sure you remove any of the residue from the bleach.
Great, thank you. Next question. Regarding balancing loads for swing bucket centrifuge when placing tubes, was the picture shown in your slide, the one on the left, an okay loading?
So this is when I'm really sorry that I can't point to things at this moment. The swinging bucket multiple 15 mil or 50 mil centrifuge tube buckets, those are like the hardest things. Again, remembering I'm showing newbies how to do it. And so if you picture upper left, upper right, lower left, lower right, so basically a little square, upper left, upper right, lower left, lower right. When you put a tube in the upper left of one of the buckets, your balance tube for that one will be in the upper left of the other bucket. And upper left is you are standing at the spindle and looking at the container. So they're diagonally across. What I tend to see people do is I think they forget that you have to like be the spindle to look at the bucket. And so they'll put it in the upper left on one bucket, but then they'll wind up putting it, I think it would be the lower left position on the other one. It's not balanced. The other thing is with those little multi-holder thingies, sometimes you have to give them a tiny little rotation so that they're directly across from each other. Bob, if you have a better way to explain three dimensions verbally, go for it.
I like the idea of the viewpoint being from the center of the rotor, but in the slide, I just looked at it. The diagram on the left is a symmetrical one, and the one on the right is the one that is an improper loading. So that really what you're looking for is just symmetry. Keep in mind that depending, let's say a dozen tubes on one side isn't necessarily symmetrical with a dozen tubes on the other side. Number one, you have to consider the volumes and the volume differences, hence the weight differences. And number two, it's the symmetry. And so you really place the tubes to be able to be symmetrical with the other side.
Okay, thank you. Next question. Do rotors need to be replaced even with no signs of damage? And if so, how often?
Good question. Replacement of a rotor is dependent on two things. Just explained, actually, cycles. How many cycles has that rotor been exposed to? And if not, if that cannot be determined, how old is it? And then from age, you estimate how many cycles. And then the other part of it is condition. So that's where the visual inspection comes in. And I believe one of the poll questions had to do with our inspecting centrifuges and rotors. This is what we do during an inspection, is we literally look down into every well or into every bucket. And we're looking, and we're doing this with magnified telescopes that are well lit to be able to look at the microcracks that might be created over time, that that are telltale signs that this rotor is starting to literally come apart. So so it's really a combination of age / cycles and condition, and from that we make a recommendation.
All right, thank you. And it looks like we have time for one more question. Is there a good book on the market that you recommend about the centrifuge techniques?
Whoa.
I can't think of one that's specifically about the centrifuge. Can you, Bob? I feel like this is why we're doing this.
We have a manual that has really not changed much for a while that we call our Rotor and Tubes User Guide. And we can certainly send it to anybody who's interested in it. It's a good overview of all the different techniques, all the different types of rotors. Actually includes a good section about the tubes. So it's, I would say, a good place to start to see if this satisfies what you're looking for. And then, of course, come to one of us in Beckman if you would like to discuss it more or need more. Excuse me.
All right. Thank you, Bob and Lori. Do you have any final comments for our audience?
Really, my only final comment is if there's any more questions, we'll try to answer them by email. But thank you, everyone, for your time and attention.
Likewise. Thank you all for attending.
Well, thank you again, Bob and Lori, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.